In this lecture series, we want to start addressing this idea of regulation of gene expression. Think about, at least, work through some of the basic ways that we might actually control what genes are turned on, what genes are turned off, what proteins are available and not available inside a particular cell. Just to give us some perspective here, think about it. We've mentioned this multiple times. Every cell in your body has every gene, right? They're all there. All the DNA is there. So obviously, if you've got somewhere around 20,000 genes in every cell, and you only need 300 or 400 or a few thousand, then you've got to be able to turn on those 3,000 or 4,000 that you need and turn off the other 15, 20,000 that you don't need. Okay, and it's obviously the gene expression determines ultimately the cell fate as well. Okay, we start out as an egg and sperm. We eventually become a zygote, formation of that, and that through the process of mitosis makes all these different cell lines, right? And whether we're making neural cells, brain cells, or cells part of our nervous system, whether we're making adipose fat cells, or whether we're making dermal cells, whatever, we have well over 200 different types of cells making up a human body, making up most mammals, and each one of these has a different level of protein production, which is guided by different genes being turned on and turned off. And so how does that occur? And just when this lecture, just kind of want to go through the sort of the basis of that type of form. And so, one of the ways we know this happens is by some modern technology known as transcriptome analysis. The transcriptome is the amount of transcripts, RNA transcripts, that can be found inside of a cell. And it became a really unique technique where we can actually go and compare different organisms or different cell types or whatever, and look and see exactly what's being produced as far as a message that's available. And that tells us what's turned on or turned off back up in the DNA itself. One of the areas that's obvious for us to see where we can see differences would be in development of from fertilization on up to formation of an embryonic form, right? And you can see here, these are 2-cell. That's the first division, second division, 4-cell, 8-cell stage. You start to have different genes turned on. A morula is about 30 cells or so before you form the blastocele, that open cavity in the middle, and then you've got all the other this is the original egg, but all the formation of all these other forms. And you can look at this expression of different genes in different areas, right? This is an oocytes itself, but and the red part means that they're upregulated, the other means that they're downregulated. Of course, with an oocyte, unfertilized eggs, most of the stuff's going to be downregulated, right? You're not going to have it in production. But as you go through these other steps, you start to turn on all those thousands and thousands of genes. And this figure D here is what's known as a heat map. And the heat map, the red stuff tells you what stuff is turned on, and the green tells you stuff is turned off. So here we are at the morula stage, and these genes in this region would be turned on because that'd be the transcripts for those genes would be there. Okay, you can see that you've got most of these are turned on or off, and then over here it reverts down. You get different genes turned on and turned off at various levels through this model, right? And basically you say, okay, whatever these transcripts are down here, and you can figure them out. They might be active or inactive. In this case, active. Here they're turned off. And these were probably ones that were used for early development, those first stages of replication and first stages of breaking, going through mitosis. Once you start to form up into little tissues that become specific, you start shutting those down and start using other genes and other proteins. Okay. Now, even once you get a full-size organism, of course, you get different expression in different organs. Your liver, your heart, everything's going to, and even between sexes. We talked about the difference between males and females as far as in mammals in particular, as far as the pressures of lactation and gestation and all those things. Well, even turns out that our muscles and things have different expression between males and females. There's about, in humans, there's approximately 400 more genes active in skeletal muscle than there at one time than there are in females. Now, of course, they've got very basic ones, housekeeping genes that all have to be turned on. But it turns out to be quite a big difference there. Always people wondered, well, muscle tone in males might be different than muscle tone in females. It turns out to be driven by some hormonal controls and other things. Kind of interesting because you're trying to, if you have an athletic function that you need to make, have a female perform more like a male or a male more like a female, then maybe there's ways to regulate that. So it's kind of a way off in this science fiction area there, but that's one of the things that you could do, and it's probably one of the things that things like, unfortunately, steroids and other things that were used various times have pushed females more toward male structures. Probably what it's doing, because those are hormones, is probably changing the transcriptomes, changing the actual production, what's turned on and turned off as far as the muscle signaling that's going on in the gene production. So it's kind of interesting. I haven't really read papers about looking at that, but I'm sure they're out there. Might be something that if you're interested in, you could look up. But anyway, the point is there are different genes turned on, not only in different tissues and different times and life stages and all that, but even sometimes between different sexes and different types. And certainly there are different environments. Even in the same sex, different environmental cues will turn on, if it's cold or hot or whatever, will turn on different gene systems and make the transcriptomes be different. Okay? Right? Now, to understand how this occurs, you got to think about, well, how do we get all this in the first place? Well, prokaryotes is pretty easy, right? You don't really have this nuclear membrane. You have the DNA there. Transcription occurs, and the transcript, the message, is immediately available to be translated. And that's exactly what happens. You get the formation of ribosomes, you get the formation of the polypeptides, and eventually proteins, almost simultaneously with the transcriptional process in these. So obviously, if you want to regulate production of protein, that's what you're trying to do. The easiest thing to do is regulate it up here at the level of transcription. Just don't let transcription occur, and you won't get the protein made if you want to turn it off. And that's the primary way that we see the process occur in prokaryotes. In eukaryotes, it's much more complicated because we have a nuclear membrane. We have splicing that needs to go on, maturing of the message, and all that sort of stuff that goes on. So each of these places is places that you can have control. And remember that eukaryotic DNA isn't just naked DNA, it's complexed up into chromatin, right, into nucleosomes and all that sort of stuff. And you have to remove that or release it in order to read it. So chromatin, controlling chromatin is one way. You can then control the transcriptional process itself. You can control the maturing process, right? Whether the caps are put on, whether the splicing does occur, how fast that occurs, etc. You can control translation because all these elements have to come together. So if they don't, you can shut it down at translation, or you can speed it up at translation. And then, of course, once it's translated, all you have is a polypeptide, and you have to make that into a functional protein. So whether it folds, allowing that folding, whether lipids are, side chains are added, whether it's all changed up, can be controlled. All right. Turns out that all these levels occur, and in fact, it's not just one stage or another, right? I mean, if you want to shut down the whole system, do it at chromatin or transcription, usually by chromatin. Okay. But every protein is regulated at each of these steps. It's a very fine-tuned process. And depending on the protein that you work with, it may be more regulation at one step than another, but it can occur. And it's an energy type thing, right? Think about it. The amount of energy required to have the protein escalates as we go through this process. Each of these steps, transcription, processing, translation, post-translation, all requires energy in some form, some catalytic activity or something to happen, right? Transcription and translation are highly expensive processes for the cell. So, unless you need that protein, if you don't need that protein for hours, then just have it turned off until you signal that you need to turn it on. That's cheap. You need that protein within seconds, heat shock proteins and other things. Maybe you want to keep it down here where it's a polypeptide, it's just not matured yet, right? It's not. You haven't popped that polypeptide into its three-dimensional form. But that's an expensive place because it's sitting around there. It's been through all these processes. Somewhere in the middle, it may be where you need something else. So you may have a message, and then you simply let the message float around in the cytoplasm until the signal comes along that you need that protein, and then you translate it really quickly and make it into the product. Okay. Obviously, if you want to go from up here, if you're stopping it at stage 1 and you want to make a protein, it's hours very often to get it made. If you stop it anywhere in between, then that time becomes less and less, but the energy demand cost to the cell actually goes up. Okay? It's a classic, supply and demand problem that you see in supply chain production. If those of you have ever had any economics can think about it, I mean, this is what things like Walmart and Amazon and everybody thinks through every day. Where do I need to put this product? Do I need to have it on the shelf here in my store, or do I need to have it back over here somewhere, or do I just not even wait about it and I'll have it manufactured when I need it? Okay, that's the same basic idea that but the cell's trying to optimize that model constantly. Okay, all right. Well, if we think about chromatin in the first stage, that's the big stopper, right? And if you shut down the system, okay, that's one of the easy ways to shut down genes is to simply silence the whole region by compacting up the DNA and making sort of this heterochromatin type area as opposed to euchromatin. There are a couple of models for that. There's at least two. One's called ATP-dependent chromatin complexing. The other is histone modifying complexes, which we've already talked about a bit. Okay. As far as the ATP ones, just to let you know what's going on there, these are units that bind up to the nucleosomes region, and in the presence of energy, ATP in their case, they'll open it up. Okay, it's called remodeling of the system. All right, and the way it works isn't by necessarily breaking apart the nucleosomes or doing anything like that, but simply moving them around. Okay, one of the ways they can move them around is you can open up the DNA and then take that nucleosome and just move it to some other spot, right? So you kind of hold it in place, but you move it. The other, which is really cool, is known as octamer sliding. And octamer sliding is once this protein complex, and again, you can see it's drawn as a complex of proteins here, and energy is applied to it, that you actually take this nucleosome, open up a little bit of DNA and slide that nucleosome down, and open up, slide that nucleosome down. It's known as sliding, and so it keeps it in place. It just uses energy to kind of just let it rotate around and move it down the system. So it's a mechanical process, and it's one of the ways we think that you can control, or in this case, you're actually opening up the DNA, making that area available for the transcriptional process. Okay, so regulating it by opening it and making it available. Okay, the other one we talked about before, but the kind of the interplay between HDAC, which is acetylating these histones, and or deacetylating them, and then HAT, which is acetylating the tails, and that allowing RNA pol to come in and take its place. And these complexes that have to do with those tails, right? Well, it turns out it's not just this acetylation that can go on. You can have methylation, you can have phosphorylation, and you can have acetylation, and all these play a different role in what's going on. In fact, they've been proposed, all these working together in different ways, have been proposed into what's known as the histone code hypothesis. And it's basically the idea that transcription is controlled by modifying these histone proteins. If you want to read the message over here, there are multiple ways that you can open up that histone set, which is called writing. Okay? And that can be done by bringing in these things that will acetylate, will methylate, or will phosphorylate. And so the enzymes come in and do that to the tails, okay, and allow them to be available. If you want to shut it down, you can and make it forms or form. You can come in and you can deacetylate it. You can demethylate it, and you can bring in phosphatases, which will do the same, dephosphorylate it. Form. And then there's reading, which you actually can make it where you can function in a different way, which is all sorts of things. There's chromatin domains. There's various things that can come in and actually play a role in sort of locking down these tails and doing things to them. So the point is, there are, under this code hypothesis, there are multiple things that can come in and actually modify that nucleosome function to make it work or not work in our forms. I don't think we need to worry too much about that, but I just wanted to point out that it's not just a simple, okay, we're going to go in and mess with the tail and it's going to fold down. Kind of your book shows it as acetylation, and if it's deacetylated, the tails fold down and hold on the DNA tightly, and you can't really get much out of it. Acetylated, that opens the tails up, and the DNA is much looser, and then it can be read and all that. And that's certainly true. Acetylating it will activate it and allow it to form. Deacetylating it will shut it down. And we certainly know that the genes, a lot of these genes that we want to turn off permanently, you've got very strong deacetylation going on in those nucleosomes, and basically the DNA is just locked around those nucleosomes. Okay, but you can do other things. You can methylate, you can phosphorylate, you can do all sorts of other things to control whether or not that gene region is actually being read. But all of it has to do with this chromatin structure and whether we are actually having chromatin there, right? In the sense that we've got this proteins complex with these histones, or whether those histones are loose and are more loosely associated with the DNA to allow the form to occur. This has ever happened? Absolutely. Your book gives a good example of Arabidopsis. Whether there's flowering or not is controlled basically in a chromatin modeling type of setup. There's a sort of a flowering gene, okay, that as long as this is in place, you don't end up having flowers. You repress flower production. As long as this gene functions and makes this protein, this activator protein, then you stop flowering. Okay? What you need to do is turn that off. And you turn it off by having another gene, this flowering gene, that comes along. And I think your book has this. This gene is acetylated over here. Okay, that destabilizes it. It allows this transcriptional process to occur. And once you make this protein, what that protein does is represses flowering. Okay, so the plant, which is most of its time, doesn't produce flowers. Okay, when it needs to flower, you have another gene somewhere else, another chromosome that is transcribed. So it's signaled that you want that gene to be active, right? And so it's signaling the signals what's going to happen here. This is what's going on. You have it transcribed. You make messenger RNA. You go through translation. You make this deacetylase enzyme. Okay, so like HDAC, if you will. Okay, that goes over and then deacetylates this region over here, this chromatin region, and compacts it back up. So it restores it. Instead of it being loose and available, it's now packed back up into its full chromatin structure. You don't get any production then of this activator protein here. No translation, no transcription, and flowering is allowed to occur. Okay? So this is classic chromatin remodeling. When the chromatin's open, and you're getting the production of this, you don't get any flowering, you shut it down, and it actually allows flowering. Sounds backwards, but flowering is an event you don't want to happen all that often, right? You want to spend all of your time producing flowers, and there's too much energy. You want to, a lot of times the plant just has to do vegetative growth to get ready to flower and all that. But during flowering time, which is controlled by environment and light cues and all sorts of things that signal this. So it's a pretty cool system, and it's all having to do, again, with chromatin remodeling. Okay? So basically turning it on, turning it off. And chromatin remodeling, shutting that chromatin down, is one of the primary ways that we just turn the genes off that we don't want. Those salivary, those glands that would produce enzymatic activity as far as digestion in my palm are turned off primarily by chromatin modeling. Those genes, wherever they are in the chromosomes, are shut down by that chromatin that's associated with them being tightly bound to the DNA. All right, that's one of the ways. Now we're going to move down those other models and talk about, okay, what about shutting it down at transcription or shutting it down at translation? Transcription's a biggie, and we're going to talk about how we can really do some regulation at transcription. That's going to take a fairly long lecture.